remember, this is very basics of genetics. And so that when we're talking about like complete dominance disorders, there's not many, but there are many genes that behave in this way that also, in addition to many, many other genes, contribute to traits. So while there are very few traits, like handedness, for example, is one, where we still believe that there are two alleles that code for your handedness, and that's it, meaning one gene for one trait. But normally we don't see this. There are a few still. So let's take a look at these. All right, so one is Huntington disease. Huntington disease, this is a very interesting one in that it is caused by a dominant allele. So this is a kind of a big problem in terms of inheritance, that if one of your parents has a dominant allele and contributes that to you, it will result in this disease. This disease is, we say it about 1 in about 20,000 people, so not super common. It's a defective protein in the brain, and by having this defective protein, it causes all kinds of neurological issues. It's very slow and progressive, meaning that bit by bit, the symptoms add up to worse and worse things. You usually see it with a bit of like loss of motor control. So for example, if when you're going to grab something to drink, you just like your brain inherently is thinking subconsciously, I'm thirsty, and then you just, grab a drink and you open it up, and you don't have to program your arm and say, hey brain, tell the arm to grab this bottle. Whereas with Huntington disease, those things start to become apparent, where somebody thinks like, I'm thirsty, and then they are looking at the bottle and nothing's happening. So that's what we mean by like loss of motor control, or you're grabbing for the bottle and you're grabbing the wrong place. Those are kind of loss of motor control issues early on. Shaking, very apparent in the hands, because we do see a lot in the hands, but people may feel like tremors or shaking in their body. One of the hardest things is personality disturbances, that people will maybe they get happier, or maybe they get meaner, or different things happen. Maybe they get more anxious about things. So we see little changes in their personality. Death is about 10 to 15 years from those first symptoms showing. This is average. It can be different depending on your health and other genetic factors that you have in your environment. And onset usually begins at about third to middle age. We usually see it. So if you are, let's say you're in your 50s and you don't know, maybe you're adopted, or your parents, perished early, you might not know that your parents were carrying one of these dominant alleles. And if you had children in your 20s, you could inevitably pass on this dominant allele without knowing that you were going to pass it on. So sometimes it will pop up in people that didn't realize they had that inheritance factor. Also, depending on a lot of things with the disease, it's all, a lot of times when we're looking at these genetic issues, it's a lot to do with what's happening on the chromosomes, and there are often many like repeats of what we call like 3 nucleotides in a sequence. And sometimes when you have a lot of those nucleotides in a sequence for a disorder at that gene, it may cause you to have symptoms like in Huntington's at an earlier age than most other people. So things can also be kind of skewed. Like I said, that first slide, a lot of it is on average. The disease for Huntington disease is located on chromosome number 4, so they know where to locate it. So that's one of the good things about it. The gene, it codes for this, like I said, this base triplicate, 3 amino acid bases, which we're going to get to a lot more in the next unit. And we see this CAG, cytosine, adenine, guanine, repeated in this area on chromosome number 4 of the DNA. I hate to say normal, but people who do not show the disorder usually have fewer copies of this base repeat. And then people who are affected will have far more. depending on, like I said before, the number of repeats that you have. So you can see there's quite a range here. The high end of not having the disorder is 34 repeats, and you have like a little bit of cushion in between. And affected people, let's say it's somebody who has only 42 of those base repeats, they may not start to show symptoms until they're in their 80s, let's say. And oftentimes there are people who have low amounts of these base repeats will die of something else before they even show symptoms of this disorder. But someone who has a lot of copies, like over 100, they may start to show signs of the disorder maybe in their like 20s or early 30s.
I was going to ask, can Huntington's be tested before symptoms?
Yes. It can. It is one of the great things that in terms of genetic testing is that, yeah, you can test for it. And so if you're somebody who has a parent who has Huntington disease, you can test for it. Here's the thing is that also is that genotypes, capital H, or capital H, small h, remember that it's a dominant allele that causes this disorder. And so just having one dominant allele will cause you to have the disorder. People who have little h, which I'm guessing all of us in this room, because it's a 1 in 20,000 on average disorder, that we would have little h, which would translate into a low amount of copies of these base repeats on our chromosome number 4. So Huntington disease, again, capital letters, passed on, functional protein, little h, meaning you do not have the disorder. Here's somebody who has a brain without the disorder, a low amount of those base repeats on chromosome 4. And here's somebody who has the disorder where they're showing higher protein levels in this base area, the main area of the brain that they call the ventricles. And then that can start to cause issues with other parts of the brain. And you can see being centralized where this protein is produced is that it can affect a lot of different areas of the brain. Another example of an autosomal dominant disorder where a dominant allele causes the disorder is called Marfan syndrome. Marfan syndrome, it is more so than anything, it affects the connective tissue, and it allows the connective tissue to stretch more so that what you see, this person, for example, her arm length is across 1, 2, 3 lockers where even a tall person might be just to here, so that all of your connective tissue, especially at your joints, stretches on each side, and her hand is 1 locker length across, whereas most of us might be like a half across. Also, other things that can happen is you can have heart issues. The aorta, which is the largest artery in your body, it's like that top artery, and then it goes like this, and it goes all the way down your body. It can be enlarged, which can cause issues, which if you take 112, you'll get into that. If you take an anatomy class, you'll get into why wouldn't something bigger be better, but it's not always. And then spine is usually curved because there's a lot of stretching of the spine, and often they have a hard time seeing far as well. And that again has something to do with the pulling of the eyeballs and connective tissue. Someone who has a functional protein, which again I'm guessing most of us in here, or all of us do, have this functional protein, so we would have the code of little m. Oftentimes when we think about genetic disorders, we often think that it is 2 recessive alleles that cause the disorder, so we're going to investigate this a bit. If you are a person who has a normal function, as opposed to having a complete dominance recessive disorder, you could be a carrier of the disorder without showing signs of the disorder, because when we're talking about complete dominance, someone who is a capital A, little a, this is like normal functioning protein, and the little a would be like a malfunctioning protein or an over functioning protein, that if you could show no signs of this disorder but carry that little a, but you mask it with your dominant allele. So again, when we're talking about complete dominance, there's no blending of the dominant and the recessive. It is the dominant masks the recessive, but then here is the danger in that you could pass on this recessive allele to your offspring. And if you get together with someone else who is also, we call it carrier, because you show the dominant, but you carry along the recessive without it being expressed, two carriers get together and they can have a 25% chance of each child having this disorder. So let's just take a look at the genetics of that. So when we take a look at the Punnett square, if you have two carriers or two heterozygous individuals, that you can see that when you do your Punnett square, that anybody with a capital allele will not show signs of the disorder. So we have a 75% chance of each child not showing this disorder when their parents are carriers, but each child will have a 25% chance of showing the disorder. So let's talk about a couple of examples of this type of inheritance. One is albinism. Albinism, it's caused by malfunction in the enzyme called tyrosinase. Tyrosinase is an enzyme that produces a pigment called melanin. Melanin is a pigment that is added to your skin. And the gene that codes for this malfunctioning tyrosinase enzyme is called the TYR gene. Individuals who are homozygous recessive, just like what we have here, little a, they will have this disorder. That means that they do not have a functioning tyrosinase gene. The TYR gene is not functioning, so it is not depositing melanin into their skin. So it's not just their skin too, it's also hair, and it's also the pigments in your eyes as well. So with the lack of having skin pigment, hair pigment, eye pigment, I mean, hair, maybe not the biggest deal. Your hair will be like whitish or yellowish, depending on your ethnicity. People of more Caucasian ethnicities, their eyes will look pink because the lack of melanin, it exposes the blood vessels in the eyes so that their eyes look reddish or pink. People of African heritage, their eyes will often be like bluish because of different genes. There's many genes that code for eye color, that people of African inheritance have another way to deposit color into the eyes, and so they often have a very light blue or glassy blue eye. And a lot of mammals, like we are mammals, or other mammals that show albinism. So you can see here's probably a wallaby. Oh yeah, it's a wallaby. A wallaby that has white fur and then pink eyes. So skin meaning white, just really, really light colored skin. Very high chance of getting sunburn. Hair white, or maybe sometimes hair is yellowish, depending on, especially on humans, depending on your ethnicity and what other genes you have. And then again, eyes look pinkish, reddish because there's no melanin to mask the blood vessels. And that's another thing that, like sunglasses and skin sun protection, very important, because without having any melanin, there's no protection for cells in the eyes or those blood vessels from the sun. And so you really need to, as someone who has albinism, they will need to have really good sun protection. Okay. If one of the two parents is homozygous dominant, capital A, even if they pair with a parent who has albinism, all their kids will receive a gene for the functional TYR gene. So here you can see that each child in this scenario, with one parent being homozygous dominant, one parent being homozygous recessive or having albinism, all the kids will have a 100% chance of being heterozygous and receiving that production of melanin. Heterozygous individuals we have here, we just did this as an example before, so same thing applies here, that if you have two heterozygous parents, or what we would call two carriers, and they mate, each child has a 25% chance of having albinism. And if you have two parents who are albino, each parent, little a, you can see that there's no dominant allele to pass on, so that the children will have 100% chance of having albinism. And hopefully that makes sense just by thinking about it. So let's answer this question here. A cross produces 915 offspring with a normal pigment and 310 with albinism. What can you conclude? So you really do need to sit down and think about percentages on this. So I'm going to give you a little hint that it would be a good thing to take 915 and 310 and add them together for a total, much like what you did when you were figuring out the percentage of cells in different phases of mitosis. So you get the total amount, and then you divide 915 by the total amount, 310 by the total amount. See what your percentages are, and that based on percentages, like is it 75%, or we're talking real world, so is it close to 75% to 25%? Is it 100% no, 100% yes, or we have another combination where it could be a parent with albinism with a heterozygous parent with a person with albinism as well. So try and figure that out. I'll give you a second. There's a bit of math here for it. It's good to remember how to do percentages, and especially when we're taking a look at these genetic Punnett squares, because really what we're synthesizing everything down to is what percentage chance do each individual have of receiving some kind of trait or disorder. Who has the exact numbers? Anybody figure out the exact numbers? It's about, right? It's about, let's say, it's about 75%. This one's about 25%. So once we know this, that if we're looking at 910 divided by 1225 is pretty close to 75%, and 310 divided by 1225 is pretty close to 25%. We're looking at all of our squares that we just did, and then we see, so this one, each individual, when you had two heterozygous parents, had a 75% chance of having normal melanin and 25% chance of having albinism. Okay. Anybody questions? All right, another example is sickle cell. Yeah, sorry. Oh, wait, say again, I'm sorry.
In terms of the regular folding, do you have to know that Lewis development makes a part of it hydrophobic instead of hydrophilic?
No. Just what we're covering here. Yeah. Yeah. Oh, sorry. Yeah, the answer was B. Thank you. Yes, to confirm. Yes, the answer was B, both parents were heterozygous. Sorry about that. All right, another example is sickle cell anemia. sickle cell anemia, it has a lot of different influences here that it's not quite as simple as I'm presenting, but there's like kind of a little bit of a, it's not purely like each child has a 75% chance of no and a 25% chance of yes, and I'm going to talk about that in just a second. So sickle cell anemia, our red blood cells in our body, they are circular in shape, and they have like a depressed area in the middle. They're evolutionarily like worked out to be favored to be circular in shape because our blood vessels are circular in shape. So, you know, just like a little game that you might have played when you were a baby where you had a cube thing and you put the different shapes, like the star in the star shape and the circle in the circle shape. That's a lot of what our body works like, is the right shapes go through. So what happens with sickle cell anemia is that the cells, instead of being round, they're kind of elongated and they have these pointy edges, like a like old-fashioned sickle that someone would use to cut wheat with, which like the Grim Reaper, if you know, like, you know, that character would carry around. People would use a sickle to cut wheat and other grains down. So it's like a big stick with a pointy end on it, and they felt like the edges looked like those sickles. So that's why they call them sickle cell anemia. Now, this type of cell works great, right? Circle cell go through circular shaped vessels. This type of shape doesn't work well with the circular type vessels because these little points get stuck in a lot of places. So everywhere you have a twist and a turn or a bend in your body, like any of your joints, those might get stuck in there. They might also pierce blood vessels, so you can get internal hemorrhaging. If you have internal hemorrhaging, meaning that it rips open, those sickle shapes rip open your blood vessels, you get internal bleeding inside. They can cause clogging of your blood vessels, that once you get an irregular surface or a tear in a blood vessel, then your immune system kicks in and it produces a clot. Those clots can affect the area that they're in, or they can dislodge, and those clots can go to other places in the body where it might be very significantly affected, like your lungs, for example, or your brain or your heart. Because they are misshapen, they don't carry iron very well. So people will suffer from anemia. Anemia will have an effect on your overall metabolism because hemoglobin, which uses iron to bond oxygen to red blood cells, will lower your amount of oxygen. So if you remember back to cellular respiration, that you want to have a high amount of oxygen to produce that more efficient kind of cellular respiration, aerobic cellular respiration, as opposed to fermentation. So this cause of anemia will also cause them to have a lower metabolic function in their cells. Infection, it causes you a low resistance to infection. Again, there's a lot of with those sickle cells, if they're constantly cleaning up cuts in the blood vessels or in the joint areas and organs, then they're not able to strictly focus as an immune system on foreign invaders because they've got a lot of cleaning up to do. The points can clog and damage organs. It can cause your liver to malfunction, causing you to have more of a yellow appearance. From high bilirubin. Liver does really, really important jobs for so many systems in our body. So it can cause a lot of other issues. In addition, it can just like cause pain, right? Imagine that you have all the time clogs and cleanup and swelling. One of the things when we have a torn surface in one of our circulatory vessels is that you get a lot of water to that area to help the immune system circulate around cleaning up that area. So you get a lot of inflammation also. I know we're hearing a lot more inflammation in the news, how that's bad. So it can cause pain in many places in your body. And then again, a dislodged clot can cause a stroke in the brain. So there's like from one little gene can cause a lot of issues. Here's the benefit. Sometimes we have these like coevolutionary factors that if you have sickle cell anemia, you are resistant to malaria. So there's one little bonus. But for most people, that's doesn't really help you if you don't live in an area that has malaria. It is most common in people of African descent. In the United States, about 8% of African Americans have sickle cell anemia or carry the gene. In Africa, it's about 20 to 40% of the population. It's also high in individuals who live in areas where malaria is high, like Mediterranean, Middle East, Central and South America, and East India. So you see it higher in those populations as well. Also, carriers, you can test for this. So if you know that it's been in your family heritage, or if you are of one of these ethnicities, then you can be tested for it too. If you suspect maybe you're having some issues, you can get a test for that. Let me just backtrack a little bit. One of the things about sickle cell anemia that is a little different than a straight complete dominance disorder is somebody who is a carrier can also show symptoms of the disease when they are under great amount of stress. So here's the thing, that having the dominant allele doesn't necessarily protect you fully, that it is a little bit different than a straight on complete dominance. Yeah.
Does that also make you resistant to malaria?
Yes, it does. It does also make you resistant to malaria. So, so that's a little bit of a good thing with a bad thing. Okay, so let's answer this question. Imagine that your friend Roger has cystic fibrosis, but his parents do not. Cystic fibrosis, let's say if, which it is, some forms of cystic fibrosis are caused by recessive alleles. They are the recessive trait. What do you know about Roger's alleles if those of his parents at the cystic fibrosis locus of their DNA? So again, we're talking about a complete dominance disorder where having too little letters or two recessive alleles causes the disorder. So here's a test cross. I'm just going to start and show you how I would approach this. Is I would first write down that it says cystic fibrosis is a recessive trait. So let's see, I'll use little c's. So somebody with a dominant allele or a capital C does not have, I'm going to use CF, cystic fibrosis, but somebody with two recessive alleles has it. So we've got Roger, we got Roger's parents, and we got mom. So we said here, Roger has cystic fibrosis. So if Roger has cystic fibrosis, I don't know why I can't talk, he will have little c, right? Okay, so let's just think about this without trying to do some big cross here. Roger has two alleles for this trait, right? Because we know we have two alleles for every trait, two alleles for every trait, two alleles for every trait. And where does he get these alleles from? From mom and dad, right? Okay, so we know that mom gave him one little c, and we know dad gave him one little c. It says Roger has cystic fibrosis, but his parents do not. So in order for his parents not to have cystic fibrosis, what must they have? Yeah. So Roger's parents, we know they have a little c, but they do not have cystic fibrosis. So then, just by thinking it through, we can see that Roger's parents are both heterozygous or carriers for the cystic fibrosis trait. and I don't know if I get into this in the notes, I can't remember, but cystic fibrosis is caused by a malfunctioning of a sodium pump in your respiratory system and your digestive system. And so what that results is, and I know a lot of times people think that cystic fibrosis is just about like a breathing issue, that not having that pump causes a lot of mucus and water to accumulate in the lungs. And it causes them to have a lot of like issues with breathing, also with getting sick, and pollen, allergies, that they can become like overwhelmed by even dust. But also the digestive system. It produces a lot of mucus, so it has very similar symptoms to things like Crohn's disease or other autoimmune disorders of the digestive system. So it's not only just lung stuff, but it's digestive system stuff. Good things about cystic fibrosis is that they're using gene therapy, and they've had many successes with it. It's not completely cured yet. They are finding that they can insert into a virus the gene for a functioning sodium channel protein, and that a person can either, they have a couple ways of administering it. Sometimes they do a tube drip down into the lungs, or they do an inhaler, and that people will, that pump, the virus will insert it into at least they're getting it into the respiratory system, and they will have normal function, but the normal function wanes or goes down over time, because when the cells replicate, they don't always replicate that gene for some reason. But they have a lot of hope because of this success that they've had. So that's some of the great things about our world today, is that smart people like you, could solve some of these problems and help people live healthier lives. So pretty cool. Okay, so let's talk about a totally different kind of inheritance, which is called incomplete dominance. So here, it's incomplete dominance. So we don't have one allele that causes the dominant trait. This goes along with Mendel's blending concept of inheritance. If you remember back Mendel's first hypothesis, he said that when he had two very different parents, like let's say red flowers, capital R, and he hypothesized white flowers, little r, that offspring would be a blend of the two, and they would have like a capital R, little r, and it would show pink. This is what complete dominance actually shows. It wasn't something that Mendel had seen in his pea plants, so he did reject this hypothesis that he made, but we did find this in other organisms. In this case, we've been talking about having two phenotypes. For example, if we're looking at cystic fibrosis, when we're talking about complete dominance, that you had the phenotype for not having cystic fibrosis and having cystic fibrosis. You didn't have something in the middle. But now here, you're going to have three phenotypes, and you're going to have one extreme, the other extreme, and then a blend of the two in the middle. So 3 genotypes and 3 phenotypes. Sometimes books tell you to do it this way, and sometimes they tell you to do it a different way. I'm just going to go through both of them in case in the future that you encounter either or a professor in the future who likes to do it one way or the other, just to know. I hate to try and just do one way and then you encounter a different way in the future and you're like, wait a minute, that's weird. Okay, so in this case, let's just say that if we're talking about, this is totally hypothetical, I made it up, that green, there's a fish, and green and yellow, combine them together and they make blue. So we're going to do capital G is green. And so we're going to use just 2 letters, and someone who has both G's will be green, someone who has both Y's will be yellow, and someone who has both a G and a Y, they're going to be blue. There's what? What about yellow? Sorry. Yeah, I'm just saying like for genes. I was just, using genes in general, but yeah. Phenotypes, if we're talking about the genotypes, it'd be YY and GG. So these are just kind of representing phenotypes, and this is representing genotypes. Oh, so we're asking if two blue fish are mated, what will the offspring be? So the way that we would do this Punnett square would be like that. I'm using all capitals just not to confuse one is dominant over the other. You could use all lowercase. Try to just be kind of consistent in my messaging. So you see that what you end up with is 1 GG in this top left box, 2 GYs, and 1 YY. And then when we translate those into our phenotypes, the GG will be green, GY will be blue, and YY will be yellow. So we end up with 1 to 2 to 1 ratio on each. So again, different than when we're talking about complete dominance, because this essentially, if we had a 1 to 2 to 1, would be a 3 to 1 phenotypic ratio for complete dominance. But we're talking about a blending thing here with incomplete dominance, and so that this blend gets its own phenotype. Anybody have questions? Here's another way that people do this. Because I'm a bit messy with my writing, I'd rather give each one a different letter. The way I would do this is curly hair. It's a curly hair versus straight hair inheritance. I would say capital C is curly, capital S is straight, and CS is wavy. Another way that people do this is they do H1 for curly, H2 for straight. So just to show you a different method. All right, let's talk about another kind of inheritance. It's called codominance. Codominance results in 1 or 2 or more dominant alleles. One of the first discoveries that they made with this was our blood types. And so they called it codominance because A and B are both dominant over O. And then they found out later that there's other traits that have more than one dominant allele. So codominance, it maybe could have been called like polydominance or something, but usually what we start with, we don't often change. The speckled chickens? No, I don't think we'd go over it. Yeah. But that is another, one of those. All right, so here's the thing about our cells that I believe I showed you a picture of this before, that when we take a look at the cell surface, our cells are not flat. Like what we often see in textbooks as very simple drawings of cells, they have this just flat surface and then they draw in the organelles and things like that. But when we take a look under an electron microscope at our cells, we see that our cells look more like a tennis ball, that they have these little fuzzy things sticking off, and all of these are different attachment sites for different molecules to stimulate something to happen in our cells or try to get into our cells. These attachment sites also will tell us information about our cells, like your cell's a Homo sapien. It will have an identifier if you are male or female. It will have an identifier of what blood type that you are. And so there's a lot of different identifiers, these what we often call like sugar molecules or glycoproteins. They can be a lot of different kinds of molecules, but these attach to things on our surface and give us information about what our cells' traits have. So A and B code for enzymes that produce a little fuzzy thing. O does not code for anything on the surface in regard to blood type. So let me just preface this with, on a person with an O blood type cell is still going to look, their cells are going to look like this. They're just going to be missing something that A and B have. They're still going to have a lot of other different attachment sites on the surface of their cells. So what they, just very simply, what our cell surface would look like for each of the different kinds of blood types, depending on which type of blood you have, and just isolating just the glycoprotein attachments for blood type, that A blood type would have, let's say that the little things that are sticking off are blue, and B blood type would be orange. O blood type would have nothing. Somebody with AB blood type would have both A and B traits on them. And let's talk about the advantages and the disadvantages of having this. Let's go back, A and B are dominant and O is recessive, and AB is the co-dominant blood type. So it shows both A and B traits, just like what you saw in the picture.
In terms of the blood sugars, if blood type A is given to a blood type B person, the white cells are going to look at the blood type A blood, realize that it's not from itself, and then start killing those red blood cells, which then cause a problem to the human body.
Correct. Yes. So what he's addressing, Joseph is addressing, is that the way that we can either get blood from someone, receive blood, get a blood transfusion, or give blood to someone, it's a little bit complicated. And that, let's say that I know I have A positive blood type, and let's say that you have O blood type and you're here and you're bleeding, and we can't get anybody to you because there's a zombie apocalypse happening right now. And so I'm the only one willing to give you my blood. Everybody else is like, no, I'm not doing it. There's a zombie apocalypse. I'm going to keep my blood. And even if I want to give someone with O blood type my blood because they need it, and we put my blood type, A positive blood type, in somebody who is O, let's say O positive even, or O blood type in general, that their body is going to respond with a crazy immune response to receiving my blood, because, and we're going to get into that, they need O blood type. They see the little attachments on my blood cells as if it were attacking their cells. It's not the same as what they, as you mentioned, their self cells have. And so you have to be very careful with blood transfusions and who receives and gives blood to whom, because a person who really needs blood could end up with not only needing the blood, but then having an immune response. And now they've got two big problems instead of just one. So if you were like, what the hell did she just say there? We're going to walk through it. Okay. So A blood type. Your genotypes for A blood type, because we have three alleles. We have A, we have B. Those are co-dominant. They both show the dominant trait. But O is recessive. And I just make the O a lowercase letter, but I make it its own thing just to show that it is recessive and something different. So somebody with A blood type, because there are two alleles for every trait, two alleles for every trait, two alleles for every trait. This is a really important place where you need to start remembering that two alleles for every trait, because A blood type could be both the dominant A's, or it could be a dominant A and a recessive O. Again, they could be OA, but I put the dominant in front of the recessive just to make my brain see that a little bit cleaner. B blood type works very similarly. A blood type is second highest in the population with 42%. B blood type, a lot less common with 10%. B blood type, dominant B could be BB, two alleles for this trait, BB, or BO, or again, OB. A blood type is the least common. It's about 4% of the population. They have to have an A and a B. So the only genotype that someone with AB blood type can have is AB. And O blood type, because any of the dominant A or B would mask O, they have to be OO. O blood type's the highest, about 44% of our population. You'll get into RH factor, the pluses and negatives. At some point, we're going to get into it too. Okay, so a person with AB blood type, a person with A blood type, They could give to anybody who has an A in their blood type. So a person with A blood type can also give to other A's. They can also give to AB blood type. AB blood type is super lucky because they can get from A, B, AB, and their dominant A and B will mask an O, so they can get blood from everybody. Anybody AB in here? That's pretty rare. AB negative is like the best. You can get from, excuse me, I should say AB positive is the best. You can get from everybody. Okay, anyway. A person with AB blood type, they can only donate to AB blood type. If they gave to a person with A blood type, the B that they have would cause the person with the A blood type getting the AB blood, it would cause them to have an immune response because they're like, I don't know what this B is. I've never seen this. Let's attack it. So same thing with B blood type. B blood type cannot get from AB blood either. AB can only give to AB. A person with O, because O is recessive, can give to everybody. Oh, who's O? Anybody O? Are you all givers in your life? You feel like is that part of your characteristics? You're generous. O can give to any blood type because it's like filler blood. The blood can be accepted. The body doesn't look at the recessive O blood type as like foreign. It just kind of is like, cool, it's recessive. I still got my dominant A, my dominant B, my dominant AB, or if you're O, you got more O. So O blood type is great. And giving. O's a universal donor. So you O people are highly wanted by the blood banks. I give blood about every 8 weeks. It's like a little community service that I find is super easy to do. You generally will save 1 to 3 lives by donating a pint of blood every 8 weeks. And it takes about an hour. For me, it's pretty close to my house. I live by UIC and I go to the American Red Cross over there, and it takes me like 10 minutes to drive there. It takes about 40 minutes to give blood, 10 minutes home. I get juice and cookies, and I could feel a little bit good about myself. And the nice thing about American Red Cross, it's nonprofit. They get blood quickly to other people. They do a lot of really great things for the world in general. Every time there's a catastrophe, they send teams of people. If you're ever thinking about just like community service in general, start with donation of blood if you can. But the American Red Cross does all kinds of great things. They can always use people to help other people. So it's a really nice organization. Anyway, recessive O will be masked by A and B, meaning A blood type, B blood type, AB blood type, and then they can also give to O. All right, now let's see who you can accept from. If you're a person with A blood, you can get other A blood, but then you can also get the O blood. You can get that. So that's kind of nice. Like for me, I'm A positive. I can get A positive, A negative, O positive, O negative. We'll get into the positive negatives soon enough. B blood type can get B and O. Who do you think AB blood type can get blood from? Okay, so A, B, O. Who else? So A, B, O, A, and B. Right, you say, okay. Yeah, so they can get from everybody. Somebody with AB blood type, they can get from everybody. Oh, poor O who can give to everybody can only receive from O. Somebody who's O negative can only receive O negative blood. So O negative blood is like really, 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 really needed. Because especially people with, O negative blood can be given to anybody, but O negative people really, it's like the only thing that they can get. So again, if you're not too afraid of needles, it really doesn't hurt. Consider giving blood. There are always shortages. Like if I'm like 1 day over my 8 weeks since my last blood donation, I will get within a day of that day, I will get 2 phone calls, 2 text messages, 3 emails that I'm saying like, hey, you're up for donation again. Please come in. So it's really critical these days. Okay, this is from the American Red Cross website, and it shows you that O can donate to all these people. A can donate to A and AB. B can donate to B and AB can only donate to AB. Okay, so here's a lot of words. Again, I like to write down this information as I go through it. So we've got a few people here. It says a couple brings home their new non-identical twin daughters, Joan and Jill. And after several months, the father begins to suspect there was a mix-up at the hospital because Jill doesn't look much like either parent or like her sister. When the twins' blood test comes back, the father calls his lawyer to start a lawsuit against the hospital. The mother, father, and Joan have A blood. Mother, father, and Joan, right? Jill has O blood. So what I just wrote down are the phenotypes of each person. I know each person is going to have 2 alleles for their genotypes, or 2 alleles for every trait. Does the father have a case? So let's figure out what we know. What do we absolutely know about Jill's genotype? She's got to be an OO. She's an O blood type. That's the only things that she can be. So we got that filled out. What do we absolutely know about Joan, mom, and dad? Just like one type of allele. They all have an A. Right, good. So we know they all have an A. Can mom and dad have a daughter with O blood type and still be A blood type? Good. So this is hypothetically a possibility. So should the dad have gone crazy, flying off the handle right away? No, right? Okay, so does this matter to us? Not in terms of this question, because let's take a look at the answers. Does father have a case? No, because parents with A blood type can't have a child with O blood type. Did we not just say that? Yeah. So that one looks good. No, because parents with any blood type can produce child with O blood type. Can an AB person have a child with O blood type? Do they have an O to pass on? No. So this is not true for right here. Yes, because this couple's children will have A blood. We just said that Joan had, or Jill has O blood, so no. Yes, because people with A blood can pass on only A alleles to their children. No, right? They can possibly be AO and pass on O's to their children. So again, A, best answer. Anybody, any questions? Okay, any questions?